Thank you all. Thank you for this lovely invitation. And I have learned so much today that I will remember a lot of things. Well, we will jump now to agriculture. I mean, from genes, now we go to tons per hectare of productions in the different part. And I concentrated in grain legumes because one of the reasons are grain legumes are the source of fantastic vitamin, proteins, and also the source of iron in a chelated form that we can absorb it without so much folic acid and all these complications from iron coming from other sources. So, and I'm going to concentrate also on the nitrogen-fixing bacteria. And the point is, everybody knows since the Green Revolution that you add nitrogen, your grain yields just pop out. There's no way, no discussion that nitrogen is the limiting factor followed by phosphorus in the soil. Now, we have another problem now. If we don't add nitrogen, the poor people, I mean the small-scale farmers, will have very low crop yields, so they will starve, but the environment will be very happy. High-input farmers will put a lot of nitrogen on the soil, and they will produce a lot of grain yield that will feed the people, but they will cause a tremendous contamination to the environment. Now I'm even more worried after listening to Dr. Radi because farmer fields are the first source of nitric oxide. So now it's not only an ozone quencher, but also it seems to be very complicated if we breathe this nitric oxide. So anyway, another problem is most of the most fertile soils are already under the cities, I mean under concrete. So whatever is left, like in Venezuela, I mean the upper part of the Venezuela where the fertile soils are, they are covered by cities. So if we just leave the neotropical savannas or the cerrado for planting, so we need a lot of input of fertilizer. So what is happening now is decades ago, people discovered a bacteria, what we call beneficial bacteria for the growth. So we have a group of alpha proteobacteria that fix the nitrogen from the air. What happened? The higher concentration of nitrogen gas is in the atmosphere, but the plants are totally unable to get it unless it's converted to ammonia or nitrate in the soil. But we have what we call the rhizobia group. It's an alpha proteobacteria, which they can form a symbiosis with the plant and fix this nitrogen from the atmosphere to cover. In the case of Brazil, in the soybean, Brazil soybean can cover until 85% of the nitrogen required for growth and production with very limited contamination. Not only that, it's a very friendly and very low costly new biotechnology. Why? Because it just costs no more than $10 at the most per hectare, and you have to add it only once. It's not nitrogen that we are talking about, hundreds of kilos at least 3 times during the crop. All right? So we have this, and just to give you an overview, what you see there, this ball, this is the nodule. You see in the root of any legume. Inside these little bulbs, which are in the root, is the bacteria. So that's what the symbiosis is. This is a structure that harbors the bacteria. So the bacteria will take the nitrogen, convert it into a nitrogen which can be recognized by the plant, be uptaken, and the plant supply all the photosynthetates, I mean organic acid, sugars, for the bacteria to survive and to carry. Signaling mechanisms are extremely complicated, and instead of learning every day is a more new molecule like lumicron from the bacteria. There are all the flavonoids, because it's a very specific recognition between these two, the bacteria and the plant. It's not so random like we thought before. What is happening is the plant is like a bride. I mean, it just sent to the environment the right. So if the right bacteria is in the surrounding, it will answer the plant with another set of signaling which is called the node factor, the nodulating factor. So the plants say, okay, here we are a good one, a good partner, and I would like to establish the symbiosis with this partner. And all right, so in the way, everything connects. And what happened is the bacteria, by a very complicated mechanism, attach to the root hair, we were talking about microns, and open a channel and go inside the plant. And in the same time, the plant is preparing the ultrastructure for the bacteria to come in and find a nice house where to live and to multiply and replicate. All right? This biotechnology is called biofertilizer. It's produced in Brazil, Argentina, and Uruguay by number of private companies, especially in Argentina, from Pergamino, Rizobacter. It's taking over Africa. It's definitely really moving and really taking over South America and even going over the Brazilian company, producing worldwide. Sorry, you maybe more, but what is happening in the green, you have crops in different countries which are being biofertilized, and there is definitely an increase. Sometimes increase can be 500 kilos per hectare, but 500 kilos per hectare for the small farmer is a lot. It's a lot of food to eat, all right? So now, more recently, people started all over the world, and in my laboratory we started to recognize that the rhizobia, the nitrogen fixer, is alone could have some problems. It requires a consortia with another set of bacteria, which I call the plant growth promoting bacteria, which are firmicutes, actinobacteria, gamma, beta, proteobacteria, a large number, but mainly they solubilize the P, the phosphorus, and they solubilize zinc, they solubilize the iron, which is in the chemical form in the soil, which is totally unable for the plant. I mean, it's enough P in the soil, but the plant cannot take it out. Okay? So, up to here, everything seemed fantastic, but I used to have a Muslim student from Ghana. He said, there's never a free lunch. The problem is, now we go to the conference of Dr. Radi. For the plants, for these bacteria, friendly bacteria, to come and form a structure inside the roots, the whole defense mechanism of the plant has to be switched off. It means the node factor, these lipopolysaccharides, which are excreted by bacteria, block all the ROS production. What happened? It says, so the plants say, this bacteria is friendly. I mean, coming inside. But at the same time, all pathogens, virus, fungi, they use the same channels, and also they come in, and they become the plants now. And this is now a real problem, because what's happening, the friendly biotechnology that we thought we solved the problem of environmental pollution by nitric oxide, and of course on the groundwaters by nitrates and nitrates and all this, now we have a problem because the plants are becoming more sensitive to virus infections. In this case, for example, we see in the left, we see a plant, any bacteria. You see how ROS is triggered in the root hair of the. But the other hand, when the friendly bacteria, so the ROS production is blocked. So, but you see, the plant, one is blocked, everything surrounding will come in. And this is what is going on. I mean, a lot of crops are showing all these symptoms of virus infections. And in the leaf is because the virus, they love chloroplast. And only that, they stop completely. I mean, they destroy the assembling of the photosystem too, because sometimes they disrupt the membrane of the nuclei, and the chromatin you can see is scattered all over, and you can see chloroplast in one of the early stages of Professor Patricia's slide before. So that's why they are like chlorotic. You see, they are pale. And all these symptoms, okay, that's a dream for a plant biologist. But I'm not so happy that it's a dream for the farmer. Production, we go back again to our main goal, which is produce food. The production, the grain production, can be up to 100%. It can be no grain. And you can imagine small farmer. It's a tragedy. I mean, the only thing they can do is just to go move out and look for another job because the farm, I mean, unless they have money to destroy, to plug again and replant again. You can see how damaged. Because another poor thing, I mean, sorry, but most of the grain legumes by infecting virus are transmitted by the seed, what we call seed-borne virus. So unless they have the money to buy a certified seed from huge companies, which are sure poor, I mean, virus-free, if they keep their own seed for the next planting, they're just keeping the virus coming over, and every time the 30%. So they're actually completely losses for the farmer. And here is an example. I mean, you have the healthy plant. You have in the middle is a plant coming from the seed-borne transmit virus. So you can imagine the farmer. There is nothing. I mean, no grain, nothing. The plants will die. And the right one is a later infection. The virus infection can come in a later stage of the growth. But in any case, it's a disaster. So this will be ameliorated if the plant were supplied by chemical nitrogen. So if we buy a chemical nitrogen, we go back to the same problem. So it's like a riddle. I mean, we protect the virus, we help the farmer to survive virus infections, but we contaminate the environment and the groundwaters. If we don't do it, if we apply the biofertilizer, the bacteria, so the plants are much more susceptible to the virus infection. And this is an example. The two upper set of A, B, C, and D, these are the regular, the normal hair roots. So you can see how when the bacteria attach, the hair root start like folding, so trap the bacteria, and it's a channel that will go into the roots. The lower one, the E photo, is from a virus-infected plant. So if I were a bacteria, I look to this root and I say, I don't go in there. So there is not going to be any nodules, no nitrogen fixation. So we go to a nitrogen deficiency. So you see, it's a really complicated. This is rose. Again, you can see the middle one, the lower one, middle one is the normal response, rose response in the radicle to infections. And the F1 is what is happening, what the bacteria, the good one, the nitrogen fixing, the rhizobia is blocking the rose production in the outer cortex of the radicle. Okay. And now, just to show you what is going on, photograph A is the inside ultrastructure of the nodules, where you can see the bacteria. These bacteria are the one fixing the nitrogen and giving back to the plant in exchange of photosynthesis. The B, look at the virus. All these massive crystals, absolutely amazing amount of crystals destroying everything, is the virus replicating. Plant viruses, they produce extra capside, so they form fantastic crystals. This is like a honeycomb. It's really beautiful. It's even a taxonomic. I see this ultrastructure and I can classify already the virus. But you see, everything is destroyed, so there is no way that this nodule will ever fix nitrogen because there is no ultrastructure. And sorry, Patricia, this same crystal, you find it attached, inside sometimes the chloroplast and destroying the ultrastructure of the chloroplast. All right? This is another view. You can see the middle one. Look at the crystal, attached. Can you see the little bit of the crystals in the upper and the lower are controls. The middle one is and in this one, in the right one of the middle, is already a totally destroyed nodule. So there is nothing will be fixed in that. This is another view of the same. So we have the positive effect of using biofertilizer is, of course, the plants have higher photosynthetic rate, higher grain yield, the xylem vessels are wider, and they can even donate to the soil up to 150 kilograms of nitrogen per hectare, and they have high rate of nitrogen fixations, and they can solubilize the phosphorus, iron, zinc. They have larger root system. Negative effects, they are more susceptible to virus infections, and the plants are more demanding on manganese, molybdenum, iron, and phosphorus, and also the bacteria is depleting the soil of microelements. So this costs to the farmer. The farmer now has to replenish these microelements. And microelements are even more expensive than NPK. So that could be another problem. Okay, now this is the end of my talk. But I want to say, because now I'm in the middle of medical doctors, so we have another critical point to see. Most of these growth-promoting bacteria are human pathogens. So I'm going to tell you, when my daughter was born, the doctor, I was afraid at the time that she will stop having milk because I knew what was coming. The doctor said, you know, you can prepare a broccoli juice, you know, in the osterizer, and I give it over my dead body. Because broccoli can have Pseudomonas, Xanthomonas, Klebsiella pneumoniae, and Pseudomonas putida have been isolated. I isolated from the broccoli, just to give you an example. So I'm going to give my daughter that just born a spoon of such horrible things. So be aware when you eat raw, because the plant, they don't discriminate very much. And all these plants, it was discovered by Esperanza Martinez Romero in Mexico. You know, she was the first one to discover that sugar cane, it was loaded with Klebsiella pneumoniae in Mexico, among other horrible things. So plants are not so healthy, and it has to be taken, especially in these farms where sewage water is used for irrigation, which is very bad, or contaminated rivers. The plants will take over everything. So you buy, good luck. Okay, thank you.